What's up, distinguished boys and girls? We will review the questions as I promised you. We will review this copy, and we will review only the questions from the first chapter. Question 1A is justify and explain the following facts. Amoeba has contractile vacuoles. We discussed this while we were discussing the organelles and the vacuoles. We said that amoeba has contractile vacuoles in order to drain the cell from the surplus water and dissolved waste. Justify and explain. The division of the adult neurons is rare. We discussed this during cell division. We said that the division of the neurons in the adults is rare because the neurons are specialized in a definitive way. Okay, I told you that the neurons in the adults are permanent cells. They do not go through cell division. But the neurons in the embryonic phases, they divide, and we said that the duration of the cell division, or the duration of the neuron division in the embryonic period, is 30 minutes. Question 2A. Complete the following. It's a blank question here. The cell wall of blue-green algae is surrounded by gelatin membrane. I discussed this in my second video, I think. I told you that this is very important. Please emphasize on it, that the cell wall of the blue-green algae is surrounded by gelatin membrane. And we said that I also gave you a question, very possible question. They will give you justify why is the blue-green algae and the typical bacteria are different in the structure, even though they belong to the same kingdom. It's as simple as because the typical bacteria do not contain chlorophyll, and the cell wall of the blue-green algae is surrounded by gelatin membrane. The cell distension, I think they mean here the cell distends and then rupture if put in blank. This is hypotonic solution. This is just basic question of osmosis here. Now let's review the pictures real quick in osmosis. Here. So this is the normal cell. It's in the isotonic solution. And the cell will distend and rupture if they are put in the hypotonic solution. And in the hypotonic solution. In the hypertonic solution, the cell will shrivel and shrink. Okay, so it's the opposite to the hypotonic solution. Very simple. Nothing much here. Question 2B. What is the location for each of the following? I really love this question. The kinetosome. Now, the kinetosome is the same as the basal body. We discussed this. So they can give you the exact same question, and they will tell you instead of the kinetosome, they will tell you basal body. It's the same structure, the same answer. The kinetosome is found at the base of the cilia and flagella in the cells that contain cilia and flagella. That's it. That's the answer. And the centromere, we discussed the centromere while we were discussing the structure of the chromosome and during cell division. We discussed the centromere. I told you that they connect the sister chromatids together, so the location is found on the chromosome. Okay? It's an acceptable answer is just to say on the chromosome. And you can also write down that it connects the two sister chromatids. Question 3A, definition question. They are telling you to define the crossing over. We discussed the crossing over during meiosis, if you remember. I told you that during crossing over from the name, it is the exchange of the genetic material between the homologous chromosomes, aka the non-sister chromatids of the homologous chromosomes. And it occurs during the packaging stage of prophase 1 of meiosis 1. Okay, very important to mention the phase and stage. So it's during the packaging stage of prophase 1, meiosis 1. Question 3b, they're telling you to mention the origin of the following. The cristae. Now, we discussed the cristae as we were discussing the mitochondria. We said that the cristae are found in the inner membrane of the mitochondria. We said that the mitochondria are surrounded by a double membrane, and the inner membrane make up the cristae, which are folds in the inner membrane. And its function is to increase the surface area of the inner membrane. You don't have to mention the function here. They're just telling you the origin. It's just the inner membrane of the mitochondria. Question 4a is telling you to answer the following question. The explanation for a mechanism of chromosome movement in the anaphase. We said that the mechanism is not fully understood, but there are two explanations. One is that the spindle filaments are thought to shrink when there is ATP. Okay, so the spindle filaments shrink in the presence of ATP, and they pull the chromosomes towards the poles. Two is that the spindle fibers form a path so that chromosomes slide on these threads toward the poles. Question 4b is telling you who's responsible for each of the following. The conversion of glucose sugar into a protein in plant cell. Very easy, really easy. It's just the leucoplast, okay? It's the colorless plastid. Question 4c is a drawing question. Draw with labeling the phagocytosis process. As you can see here. Again, any drawing question is just a free question in my opinion. Question 5a. Comparison question. Compare the differences between mitosis and meiosis. So, what are we going to say here is that the main points that we discussed during mitosis and meiosis, I kept telling you that during mitosis, only one cellular division is happening. But during meiosis, it's two divisions. We have meiosis 1 and meiosis 2. The final result of mitosis is two identical cells. But during meiosis, it's four non-identical cells. During mitosis, they are genetically identical cells, but during meiosis, they are genetically different. Of course, the number of chromosomes in mitosis is similar to the mother cell, they're equal, but the number of chromosomes in the new cells is only half the number in the mother cells, so we get haploid. In mitosis takes place in the somatic cells, but meiosis takes place in the reproductive cells. Mitosis division occurs during cell life cycle continuously, but meiosis division occurs after sexual maturity. And the mitosis division is used for growth repair cells and asexual reproduction, but in the meiosis, it is actually used in the sexual reproduction and having new members of the species. Question 5b, give an example for each. The metamorphosis process. It's only one process. It's only one example mentioned in the book. It's just the frog larvae tail that disappear. Question 6a. They're telling you to mention the function for each. the dictyosome. Again, the dictyosome is just the Golgi apparatus in the plant cells. And the function of it builds cellulose, okay, and some components of the cell wall. And question 6b, it says, what are these sentences indicate has a vital role in the formation of ribosomes? What is it? It's, of course, the nucleolus.